Hi everyone, welcome back. So last time we talked a lot about viral anatomy and how viruses make more of themselves, this viral life cycle. The gist of it is that all viruses have to find a way to infect a potential host, infect a cell of that host, and then hijack its many resources and tools to turn that cell into a viral factory, to make it make more viruses. All the while, it has to avoid being destroyed or discovered by the cell or the organism's immune system. And eventually, when it has made enough little viruses or the virions, it has to escape the cell and then find new hosts to continue that cycle, either through destroying the cell, like lysis, or from budding off from the cell, different things like that. Along the way, these viruses are going to be replicating their genome, or more accurately, it's going to force the cell to copy the virus's genome, either the DNA that makes up the virus or the RNA that makes up the virus genome, depending on which type of virus we're talking about. And so, anytime that the genome is replicated, there is an opportunity to not do that perfectly, to introduce an error. And those small errors, those improper or imperfect copying of genetic material, that is what a mutation is. So yes, viruses can also experience mutation just like living things can. And these mutations create variation. They change the DNA or RNA of this virus, which can result in a change to the characteristics of that virus, how it interacts with its potential hosts, how it forces it to replicate itself, how it survives in the environment in between hosts. So these mutations could have positive effects for the virus. They could have negative effects for the virus. More often than not, they have neutral effects, so they don't really have much impact at all. But importantly, some of those mutations can have real impacts for viral success, for the ability for that virus to infect something new or use it to replicate. So these new mutations might create opportunities for the virus, like making it less recognizable to a host's immune system, for example, or it might give that virus access to a new host, a different species, or to make it more effective at infecting a particular host species. These viruses all have a set collection of host species that they can infect. These are what we call the target species, host species that the viruses have evolved to infect best. And oftentimes these viruses can be very specific, where they can only really target one or a few different species. There are, for example, some viruses that we know only infect humans and some primates, which are our close relatives. And that's the kind of thing that we often expect for a lot of viruses, although that is not the case uniformly. Now, while these mutations that we introduce can create new traits that can allow the virus to do new things, another force called selection favors whichever of those traits are best. And by best, I mean which ones help the virus succeed, help it infect a new host, help it replicate itself within that new host, help it spread to new hosts. So selection is essentially saying those viruses that are successful reproduce more, and then they pass on their genetic information easier and more often than those viruses that are not as successful, that don't replicate as much. And so the population of viruses steadily shifts towards being more and more like the successful viruses, the ones that are actually able to reproduce. As they do, viruses often become more specialized. They become more effective and efficient at infecting or utilizing specific hosts, very targeted host species. That said, it is also possible and happens periodically for viral infections to what we call spill over into non-target species, things that the virus did not evolve initially to infect. This can happen because the virus may undergo a mutation, like we mentioned before, and that changes the way that it interacts with potential hosts. Suddenly, its unique features might be now a better fit to infect that new species rather than its previous species. This change, of course, is not intentional. Viruses don't have brains, they don't have intent, there's no will behind any of this. It just happens to be a mutation that worked out for that particular virus. It's important to keep that in mind. These instances where viruses spill over into new hosts, these are what we call zoonotic events. Essentially, this is just an opportunity for a virus to switch hosts, to infect something new, moving from one species to another. Sometimes these zoonotic events aren't even successful, where the new host is very effective at fighting that virus off. But we still call these things zoonotic events because it's an instance of that virus infecting a new host that is not the target species. But if that zoonotic event is successful and the virus does very well in that host, it replicates a lot, and then whatever the mutations were that allowed this virus to infect that new host could allow that virus to spread to new hosts of that same species. And that can create the opportunity for that virus to thrive in that new host. Of course, what species are viable and effective for this virus is not always necessarily black and white. It's not always the case where we have a set of highly effective species for a virus to infect, and then a bunch of other species that the virus simply can't affect, and it requires some kind of unique mutation in order to get access to them. Sometimes it's just kind of a scale where viruses can sometimes naturally just overlap and spill over without some new mutation into a non-target species. So when that happens, these viruses are infecting, what we think of as unideal hosts. And the virus isn't as successful in those new species as they are in their target species. That doesn't mean they can't necessarily replicate and effectively use their host to make more of themselves. It just means that they are probably more effective in other hosts. So a really good example of this is cowpox. So cowpox is a viral disease that infects cattle, but can sometimes spill over into humans as well. And it does this because the virus is quite closely related to a human disease called smallpox. Thankfully, smallpox is gone today, but cowpox still exists. And historically, because these are such closely related diseases, the viruses are very closely related to one another, they can still infect similar hosts. And so occasionally, even though cowpox is evolved to infect cattle, it can more easily spill over into humans rather than other mammals because of the similarity in the viruses and the mechanisms that they use to infect us. So humans here in this situation are not really an ideal host. It doesn't replicate as well in humans, but it doesn't mean that it can't do that at all. And so sometimes this spillover does happen. So this periodic zoonotic spillover was actually kind of useful in a weird way to humans, though, because humans were plagued by smallpox for millennia, and smallpox was a really devastating disease. It had very high mortality rates, and if you contracted it and you survived, it would still really scar your body. But there was an interesting observation for people that contracted cowpox. We later realized that people who contracted cowpox, because of the virus's genetic similarity to smallpox, had immunity to smallpox. Edward Jenner is the person who was famous for making this realization and putting it into practice. He realized that these zoonotic cases of cowpox could grant immunity to a much more dangerous smallpox. And so he put this into practice in 1796. The practice of variolation was something that was already done at this time and for millennia before this, which is the process of taking infectious material from a smallpox patient, someone who actually had smallpox, taking like pus from the infected areas and scraping it onto the skin of a healthy person to give them a controlled case of smallpox that would be safer in a lot of cases and less likely to result in death or maiming and things like that. Well, this new method, vaccination, was even safer because now, instead of exposing somebody to a deadly disease, they were exposing somebody to a closely related, far less deadly disease, cowpox. This is very early vaccination, this inoculation practice for stimulating the immunity to a disease that now, today, of course, we use the term to describe all kinds of inoculations for that purpose. So I also wanted to talk about retroviruses here, because retroviruses are really peculiar. We've talked previously about how we have different types of viruses. Aside from naked and envelope viruses, we have DNA and RNA viruses, where their genomes are based on DNA or RNA respectively. Well, some of these RNA viruses are also retroviruses. This is a type of virus that does something a little bit odd. It takes their genetic information and incorporates it directly into the genome of their host. And in doing so, they force it to replicate that viral genome essentially indefinitely. This is a really interesting quality because, remember, this is an RNA virus. And so you have to ask a question, okay, it's infecting living things that have DNA-based genomes. How is this virus going to take its RNA genome and insert it into the DNA genome? Those are not directly compatible. Well, to accomplish this, retroviruses have a really cool special enzyme. It's called reverse transcriptase, okay? An enzyme that takes the RNA of that virus and makes a copy of it as double-stranded DNA. And so when it does, it can insert that copy directly into the host's DNA genome. Now they match. This should remind you of the bacteriophages that we talked about last time, where they have the lysogenic phase, where these viruses can insert their genomes into bacterial genomes and use those to copy and replicate themselves basically indefinitely until those cells become stressed and then are forced to express those viral traits again, being turned into viral factories. This is a very similar process. Retroviruses are not bacteriophages, but they do have this quality in common. And there is at least one major prominent example of a retrovirus that infects humans. There are retroviruses that have infected living things throughout history and still do today across the diversity of life. But for humans, the one that we really think a lot about nowadays is HIV. So retroviruses create really problematic, long-lasting infections. They create the conditions for what we call a latent infection. This is an infection that can persist a long time without manifesting symptoms. Organisms who are infected by a retrovirus may live out their lives without really realizing that they have that infection. Then, after some period of time of incubation or residence in the cells, it can emerge as a symptomatic infection. Now, these latent infections aren't exclusive to retroviruses. An example of one that's not a retrovirus is like chickenpox and shingles. When it infects you the first time, it creates a problematic pox infection. It can be very painful and itchy and difficult to deal with. But most people who get chickenpox can get over chickenpox. When the virus infects you the first time, it finds its way into nervous tissue and then goes dormant for a long time. Only during periods of, for example, stress or weakened immune system or things like that can the hiding virus then emerge and become shingles, expressing itself in a different way. That is an example of a non-retrovirus latent infection. It doesn't do anything with your DNA. The virus just stays there for a long time. But some latent infections are retroviruses, like HIV. HIV will infect cells and then hide in there in the genome for a long time. This is a pretty significant difference between other types of infection, like, for example, an acute infection. This is, think about like any time that you've had to deal with the flu, where it's a momentary or short-lived infection, where you battle it for a little bit, it lasts maybe a week or two, and then you get over it and the virus is gone, the infection is over. It's not coming back unless you encounter it again. And then you have persistent or chronic infections, and these are infections that will persist after that primary infection. Hepatitis is a good example of this, like hepatitis C, for example. The initial infection lingers and it stays there continuously until you either are able to fight it off, oftentimes through medication, usually exclusively through medication, or you die. A latent infection could also be something that is slow to build. This is when you have an initial infection, a spike, and then a waning effect, and then a slow ramping over time. This is the pattern for HIV, where your initial infection may have physical symptoms that impact you, and then over time that gets better, but then you have the retrovirus in your system that starts to accumulate and cause problems steadily over time. It steadily ramps and ramps and ramps, and then eventually it can transition into a full AIDS infection. It's the same infection, it's just the effect of time that has allowed it to manifest as AIDS rather than HIV. This we typically call a slow infection. It is a latent infection, but it's a type of latent infection. So latent infections in retroviruses can come from changes to the gene expression of these infected cells. When a cell is infected with a retrovirus, it has had viral DNA inserted into that cell. And when that happens, the viral DNA is not necessarily being expressed. The cell isn't at that point a viral factory being forced to build all the little components of that virus. It may actually go dormant for some time, where the cell shuts off those genes and prevents them from being read. However, over time, gene expression can shift. A cell can forget that it silenced those genes, that it turned them off, and reactivate them. Sometimes this happens due to stress. And then those genes can become unsilenced and expressed, in this case turning the cell back into this viral factory where it's forced to make viral components and build more virions. So this whole retrovirus thing, this inserting genetic information into our cells and then allowing cells to go about their daily lives, replicating them, well, that is an example of something that we call horizontal gene transfer. In this case, coming from viruses instead of from some other source. It's important to remember that once this retrovirus infects an organism's cells, even if that organism recovers from that infection, those genes stay with those cells. Even if those genes are silenced and they never reappear in an organism that's been infected, the damage is done in a sense. The DNA is part of you. And so usually this is something that occurs inside of our somatic cells, our body cells, the cells that are not destined to make egg or sperm cells and therefore become, a future human. But that doesn't mean that that can't happen. Sometimes these infections can occur in these specific cell types, these reproductive cells, the meiotic cells. And when that happens, we create the opportunity to pass on this genetic information from a virus into our offspring. Now, it's important to remember this is only possible with meiotic cells. Those are the ones that could potentially become a new human. That is not something that is possible for somatic cells because they don't get inherited. Your skin cells, for example, stay with you your entire life, and eventually when you die, they die with you. Your gametes, those meiotic cells, could potentially, if you had kids, they go on to become a new creature that then has part of your DNA. If that cell had the DNA of that virus, then that person now also has the DNA of that virus. In this way, we can have instances where horizontal gene transfer, as in any kind of passing of DNA that is not from parent to child. We can have that kind of horizontal gene transfer transition into vertical gene transfer, moving from parent to child in that typical generational pattern. This influence from viruses, you may not think this happens all that often, and, on an individual basis, we don't really expect it to happen to any one of us, but across time, across history of humanity and the species that existed before us, our ancestors, this has actually happened numerous times. And so we know today that about 8% of our human genome is viral in origin, which is kind of crazy to think about. 8%. Now, of course, that doesn't mean that you are walking around like a living time bomb. Like at any moment you could, get super stressed and start making viruses from 10 million years ago that your ancestors picked up. No. Mutations have occurred over millions and millions of years that have taken those viral genomes and changed them into other things. They have broken them down into non-functional pieces of DNA or changed their function into something new. There's actually, in a weird way, an evolutionary benefit to this kind of horizontal gene transfer, moving DNA from viruses into animals like us. It's an interesting way of suddenly acquiring a lot of genetic information, new genes that suddenly show up in the genome. Normally, that doesn't happen through typical mutation. And these new genes, even though they start out as something that's very destructive, can undergo mutations and change and eventually become something very useful. Researchers today believe, for example, that the placenta that occurs in placental mammals is a feature that came about through repurposed viral genetics.